Okay, so welcome everybody to the second to the last ENST seminar for the spring. Today we are going to hear, it's my pleasure to introduce, and we're going to hear from Alyssa Wellman Houde. So Alyssa joined my lab about a year and a half ago, but I've known her throughout her time here at University of Maryland. She is a UMBC graduate and did some work in urban trees initially, if I remember correctly, and then moved into doing some work in agriculture and worked and participated with my lab group, and has been working over basically through COVID, trying to measure nitrogens, trying to get a better handle on nitrous oxide pathways in agricultural systems. And so that's what she's going to be talking to you about today. Alyssa has been a champion in the lab, so she's had to do a lot of this on her own in the middle of COVID. And on top of that, we have, as some of you might not know this is a huge issue in our world. In molecular biology, everybody's doing COVID tests right now. And so there is a world pipette shortage. And so we've limped along with her being very creative in how she uses pipettes to make sure that we have enough. So that's like taking a master's and then adding a whole nother level of problem when you're not sure if you're going to be able to source the main thing that you need to do your work. In addition to being a scientist, some of you may not know this, Alyssa's also an actress. And so I had the pleasure to go and see her perform in Baltimore shortly before COVID. And during the pandemic, she's done some online theater, and we're hoping that we get to see her in person again soon. So with that, I will go ahead and let you get started, Alyssa.
Okay, thank you so much for the introduction. So my talk today is titled Investigating the Effect of Management on Agricultural Organic Nitrogen Cycling and Nitrification Pathways. And as Dr. Yarwood was saying, we are doing this in the context of nitrous oxide emissions. So this is a figure taken from the EPA, and nitrous oxide is a major greenhouse gas accounting for about 7% of total greenhouse gas emissions as of 2019. And while there was a little bit of a decline in the early 90s with the passage of different vehicle emission standards, it has stayed pretty constant and has been slowly increasing again over the past 30-ish years. It is primarily responsible for ozone depletion and has a large radiative force, about 300 times more than carbon dioxide. Looking at it by source, while there are many different potential sources, you can kind of see in the diagram here, wastewater treatment, transportation, again, those vehicle emissions all playing into it. 75% of that nitrous oxide is due to agricultural soil management. So that in turn is due to soil nitrogen cycling and taking a closer look at nitrification and denitrification in these agricultural soils. So what does that look like? This is a simplified version of the nitrogen cycle with a highlight on nitrous oxide in agricultural systems. So if you look, there are a couple different inputs. We have atmospheric nitrogen being fixed. We also have soil organic matter decomposing and being a source of nitrogen in these systems. There's also fertilizer inputs, which is fairly common in a lot of agricultural systems. So what controls nitrous oxides? Well, soil conditions are a major player. Temperature, increasing temperature can increase mineralization, especially of that soil organic matter, which can then increase the nitrous oxide emissions, especially from nitrification. Soil moisture can have a similar kind of effect on mineralization, as well as can act as a priming effect on soil organic matter. So nitrification is an aerobic process, but if you have studies have seen if you have about 60% of the water-filled pore space, so right at that threshold, it can act as a way to increase the decomposition and mineralization of these organic nitrogen sources while still maintaining an aerobic system, and we can see spikes of those nitrous oxide emissions. Soil moisture itself, as that process dictates, can then have a dominant effect on how we regulate and notice nitrous oxide emissions. Again, nitrification being anaerobic and denitrification being anaerobic. So the percentage of water in a soil can determine whether or not nitrification is going to be the major source of nitrous oxide emissions, or denitrification might be. Also under soil conditions are pH. So the lower the pH tends to increase nitrous oxide emissions. You have a graph over here, and while there's some variability, you can see that there is an inverse relationship between the two. And there has been some research into some potential mechanisms as to what might cause this, although more is needed. We can see that pH might affect the enzyme synthesis. So enzymes that reduce nitrous oxide and help it continue along the nitrification process might be hindered, as well as pH shifts can cause the microbial community itself to shift, causing an imbalance. There are also management strategies that can have an impact. So fertilization across the board has been seen to impact nitrous oxide emissions and cause increases, whether it is organic, synthetic, fertilizers. Fertilizer inputs tend to lead to increases in nitrous oxide. That said, looking at the different kinds, organic fertilizers can have positive impacts on the functional microbial growth. Synthetic fertilizers can lead to acidification. So again, going back to that pH relationship can be an impact, lowering the pH, and then again, increasing nitrous oxide emissions that way as well. So there's been further research into enhanced efficiency fertilizers. So polymer coated urea or different things that are going to slow down the rate of nitrification. There are also research into different amendments that can be added to alter these processes. So biochar, a lot of biochar application studies have shown that it can decrease nitrous oxide emissions and has a relationship with the actual microbial community. Also, nitrification inhibitors can target microbes on the earlier part of the microbial community, those that oxidize ammonia, slowing that down and thus decreasing nitrous oxide emissions. That said, nitrification inhibitors can be toxic. So a lot of studies have shown, at least earlier nitrification inhibitors, in order for them to be effective, they needed to be applied at higher rates. And this in turn can be toxic to plants. Other studies have shown these nitrification inhibitors, these chemicals, in the milk of dairy cows, so they can have a long lifespan in the system itself. So again, going back to this nitrification, denitrification system, what we are studying is looking at functional genes. So how can we understand these different fluxes in relation to different microbial groups by looking at specific genes that code for specific enzymes. So these can be indicative, they can be used to determine soil chemistry mechanisms. We can get a better idea of the microbial communities by looking at specific genes. So kind of walking through an example of what that might be. AMOA is a gene that codes for ammonia monooxygenase. So anytime you see one of these things in the presentation from here on forward, if it's in italics, that's generally referring to the gene itself that is encoding for this specific enzyme, which then again, we can note that ammonia monooxygenase is specifically related to ammonia oxidizing archaea. So by taking a look at the AMOA abundance in different samples, we can then correlate that to what is going on with specific fragments of the microbial community. So how do we do this? Well, different genes are associated with different steps of nitrification and denitrification. So a little bit of background information with that. Nitrification has multiple steps to it. The first one being ammonia oxidation, in which ammonia is oxidized to nitrite. And this is primarily mediated by both bacterial communities and archaea communities. The bacterial communities, there are multiple genera involved, but primarily Nitrosomonas and Nitrosospira. And here's a diagram of what that looks like on an even smaller scale. So the genes we look for with this particular transformation are looking again at that AMOA that I talked about previously. In this case, it would be AOB because we're talking about bacteria. And that is the rate limiting step here. So ammonia monooxygenase takes the ammonia and turns it into hydroxylamine, which then in turn can be turned into nitrite. There are other functional genes that are in here that are traditionally thought of with denitrifying communities as well. That said, we don't tend to target those when we're trying to get an idea of this specific community. We tend to look at that AMOA gene. Similarly, as I mentioned, it can be present in archaea as well. Very similar diagram, although slightly different. You'll note that it didn't have that NOR gene present. But again, AMOA is what we want to focus on here. The second step being nitrite oxidation. So that is taking the nitrite and turning it into nitrate that the plants can use and assimilate. So this, unlike the previous step, which can be mediated by both archaea and bacteria, this is just bacterial species that we know of, focusing on the genera of Nitrobacter and Nitrospira. And again, a diagram showing this. The functional gene we focus on here in order to categorize it is NXR. So that codes for nitrate oxidoreductase. And we can look at two different subunits of this enzyme and genes that code for those two different subunits to really focus on two different species here. So subunit A, or NXRA, is going to code for Nitrobacter versus NXRB is going to code for Nitrospira. In recent years, there have been new understandings of nitrification. So those two steps are what we have studied for years and years. But as of 2015, we have noted something called Commomox nitrospira. It was discovered at a couple different wastewater treatment plants and also in some biofilms accumulated at aquaculture centers. And currently we know that there are two clades of them. And they're based on differences in AMOA between the two different kinds of clades. And we are slowly but surely starting to understand the impact on potential agriculture. But why are these important? They're interesting because they can do it all. So you can see a diagram here of an artistic rendering of what the different enzymes look like in the cell itself. And you'll note that Commomox nitrospira contains genes that produces all of the enzymes needed to completely oxidize ammonia from ammonia to nitrate. So this changes how we think. This changes how we think about nitrification and how we might understand how nitrous oxide is getting into the environment, because we think of this nitrous oxide emission in relation to nitrification as a leaky pipe of sorts. It comes out when these two communities don't quite line up. So if we can see that there are communities or species of microbes that are doing it all, that will affect how we look at this in the future moving forward. And you can see that here. The other communities we want to look at and the other half of the process is denitrification. So denitrification, it is more reasonable and helpful to look at it from a functional gene perspective because of the sheer diversity of species that are potentially responsible for denitrification in soils and in agricultural systems in general. Estimates say about between 1 to 5% of microbial populations are denitrifiers. And again, you can see the breakdown here of at least 344 total species in some of these agricultural soil samples. So what we look at, again, are those genes related to the different transformations. So the first one being nitrate back to nitrite again, and this is mediated by the NAR gene or NAR, which codes for nitrate reductase. Again, this is an anaerobic system, so a lot of these genes are going to be inhibited by the presence of oxygen, and this is common in many bacteria. The next transformation being nitrite reduction, where it is going from nitrite to nitric oxide, and this is mediated by the NER gene, which is coding for nitrite reductase. This in turn has two different forms based on the actual enzyme itself, one containing copper, while the other one having a heme or an iron form. These two different genes code for two different portions of the population, which in turn have two different niches in which they live and do better in. So you see differences in these based on environmental factors and sampling location often. The next step is looking at nitric oxide being reduced to nitrous oxide. This is mediated by NOR, which is the nitric oxide reductase. This is often where nitrogen-nitrogen bonds are formed in nature. And this is often a short step. It occurs pretty quickly because this nitric oxide is toxic to the cell internally. And then the final step, having that nitrous oxide being reduced to nitrogen. And this is mediated by NOS-Z. So this is the nitric oxide reductase, and it is again typically found in bacteria, capable of complete nitrification, and as with all of these other enzymes, it is strongly inhibited by the presence of oxygen. So, one last look at the nitrogen cycle before we move forward. That was a lot of information, and you can see how all of these different steps again play into it, and where we can see some of this nitrous oxide escaping into the atmosphere at large. How does this play into the different functional genes? Well, looking at some of the past literature, that NOS-Z gene, which is responsible for that last transformation, there have been studies that have shown increases in nitrous oxide emissions when it decreases, which makes sense to a certain extent. If you were to have a build-up of this nitrous oxide and a lower population of bacteria containing this NOS-Z gene, it is going to be less capable of turning that nitrous oxide into atmospheric nitrogen, and some of that nitrous oxide might escape. Another thing that we have looked at in the past are NRK and NRS, with the idea that if the populations of those increase, it is going to increase the second half of this denitrification. So it would create a downstream effect of increasing the nitric oxide, which would in turn increase the nitrous oxide, which will build up in the soil system and potentially escape into the atmosphere. Looking at the two together, we see a relationship where if we look at the ratio of these early denitrification genes to this later denitrification gene, we see if it is a higher proportion of the earlier denitrification genes, so an increase in that ratio, we see an increase in nitrous oxide emissions. Following that same logic of if we increase this, there's a downstream effect. That NOS-Z gene, it's still about the same population or a smaller population. That's something that's going on earlier upstream. So we see nitrous oxide being emitted into the atmosphere. So those were the main things that we've seen a lot in previous literature studies of denitrification. And a lot of the prior research in this field has focused on denitrification. Denitrification has been thought of the primary way that nitrous oxide in agriculture occurs. That said, more recent studies have shown that nitrification might also have an impact. It might be a significant source of nitrous oxide. So some of the studies have looked at AMOA. So again, that is the gene that codes for both bacteria and archaea that oxidize ammonia. And they've seen mixed results across the board, but leaning a little bit more towards if there is an increase in this gene, we tend to see increases of N2O emissions. Which again makes sense. If you are taking more ammonia and you have more populations ready to turn that downstream into nitrate, you are also then potentially creating more nitrous oxide, especially if there are less abundant communities down the line. The other community there being that NXR gene, if that remains stable, there will be more buildup earlier than what the gene in the population later on in that process is able to accommodate. There's been some research into how management has affected these different functional genes. So nitrification inhibitors, again, they target those ammonia oxidizing bacteria. And so looking at the previous slide, we can see that that can have an impact on the nitrous oxide emissions. So if the AOBs decrease, we tend to see a decrease in nitrous oxide emissions. Also, again, adding additional N fertilizer. So any sort of nitrogen inputs have been seen to increase a lot of nitrogen cycling genes, those AOBs, as well as those denitrification genes, NRS, NRK, and NOS-Z. Generally, the AOB community has been noticed to be more sensitive to change than the archaeal community. And we've also looked at manure additions. So understanding that synthetic fertilizer can cause this change, but trying to understand how different sources of nitrogen might impact this. And they've seen that manure, which will break down a little bit slower than some of the synthetic fertilizers might, increases those nosine genes down the line, which in turn also decreases the AOB potential early on and has seen an overall decrease in nitrous oxide emissions, one study reporting it a decrease by about 14%. So our project was specifically looking at these different alternative nitrification and denitrification pathways with the goal that we want to be able to improve predictions and reduce these nitrous oxide fluxes by understanding that microbial community. So our hypothesis was that the NRK and NRS, the two denitrification genes, and NXRA, which is the nitrite oxidizing nitrobacter functional gene, will be higher in the no-till and organic soils when compared to conventionally till soils. So the project itself had a couple different study sites, the first one being at the Farming Systems Project in Beltsville, partnering with some collaborators at the USDA. The plots have been long-term established since 1996. Some climate data there for you, very similar to what we would expect in College Park as well, and the soil type. There were 4 different broad categories of soil type, but generally all fell under the category of silt loam. And we looked at several different treatments from these sites. So we picked a subset of plots, 3 of which were no-till, 3 of which were chisel-till, and 3 of which were organic 3-year rotations. And you can see some of the different treatment differences between these different management practices. So between the till and the no-till, crop rotation, similar. The main difference there being the tillage, weed control, and fertilizer, similar, both receiving a synthetic nitrogen supplement, versus the organic 3-year rotation had a slightly different crop rotation, was also tilled, but weed control was cultural, so they did not receive any herbicides, and the fertilizer input was primarily manure-based. A little bit of background about the Beltsville site. We are still hoping to, in the future, pair some of this to the nitrous oxide data. This presentation is primarily focusing on the different functional gene communities, but a little bit of context, these are sites with longstanding nitrous oxide emission data. And there have been studies in the past that have looked at a little bit of the difference between the no-till, chisel-till, and organic sites. Generally at these plots, they've seen that the organic has higher than the no-till. They didn't necessarily see any significant differences between the chisel-till from the organic and no-till, and there were significant differences though between the different systems in the nitrous oxide fluxes. The other sites that we were working with were with collaborators from the University of Minnesota, and there were 3 sample sites taken from around the Minneapolis region. Again, a little bit of climate data here for you. They were similarly under a mixture of management techniques, and we picked these sites for 2 reasons. One, again, there was corresponding nitrous oxide data that we hope to incorporate moving forward, and there was also a history of nitrification research at these sites. So as I mentioned previously, denitrification has been the focus of research, but in years we've seen a shift towards nitrification, and these sites have been included in a lot of those studies. The first one being the Sand Plain Research Farm in Becker, Minnesota. Soil type is loamy sand. It was under a rye potato / soy wheat rotation, so a little bit different. And the plots we were looking at were control plots, which received no fertilizer the year where the samples were gathered. They were chiseled and disc plowed, so conventionally tilled. And again, for context, a little bit of the nitrous oxide data there. The 2 treatments we looked at were one that was taken in 2018. So these were archival samples that had been air dried and sieved. And then a sample that was fresh taken in 2019 post-harvest. We also took samples from the St. Paul Research and Outreach Center. These soils being silt loam, and this under continuous corn since 1996. So again, pretty comparable to what we are seeing in Beltsville. And similar to the previous site, no fertilizer applied here and conventionally tilled. A little bit of nitrous oxide data for context. So higher than the Becker site, but still fairly similar to what we were seeing at the other site in University of Minnesota at Rosemount. So this is a silt loam soil with corn soy rotation, again, long-standing. This one did receive fertilizer as urea, and we see similar, slightly lower, but similar nitrous oxide emissions historically from this site as the previous one. And the treatments we were looking at here were two different sets. So we saw some plots that were no-till, and then we looked at some plots that were conventionally tilled, a mixture of moldboard and chisel till, depending on what year it was harvest. All of these were taken post-corn harvest. So as I mentioned previously, these sites were all part of longstanding nitrification studies. And some of the things they had seen in the past was that Nitrobacter, so that NXRA gene, had a regulatory force over N2O emissions versus the other NXR gene, NXRB, which codes for the genus Nitrospira, was more impacted by management types. They looked at the ratio between NXRA and AMOA, so those two different nitrification communities, and saw that Nitrobacter, it can be suppressed due to ammonia toxicity, and this is all current ongoing research. So once we had these soil samples were 0 to 10 centimeter composite soil samples. They were immediately stored at a -40°C freezer to keep them preserved. DNA was extracted and quantified using a qubit fluorometer. We looked for the different genes' presence and absence, as well as tested to see if the primers we were using to isolate these samples were effective using PCR and gel electrophoresis. We cloned different standards for some of these samples for qPCR analysis using E. coli as a vector and a topo cloning kit. With the idea that ultimately we wanted to be able to compare qPCR data between these different samples. So qPCR is used to determine gene copy number. And the way that it works is you have a series of standards with known gene copy numbers. That's why we were cloning it before, was to develop those standards and then be able to dilute from that stock down to, again, a series of known standards. And how this works is there's a binding dye included in the master mix. We use Cyber Green, and it fluoresces with increased amplification as the qPCR process goes on through a series of thermocycling. The targeted gene will amplify, and there's a detection threshold established in the machine. So what you see when you see these graphs is a CT value over here, which is indicating a threshold cycle, which is the cycle at which the fluorescence is detected. So the CT value is inversely proportional with the amount of whatever we are looking at here. So for instance, something with a CT value, all of here, the samples were along the lower part of the standard curve, which is to be expected of a functional gene. This is the raw value from one of the NERK runs that we did. You see that the lower values would potentially have the CT value of about that 20 versus something that would have a greater abundance would be something more around that 18 value. All of this are then turned into the count values, or the CT values, which we then turn into counts so that we can actually compare the gene copy numbers. So these are the different functional genes which we were hoping to look at this time around. Some of these are still in process. We've had some trouble isolating the NXRB population as well as the Comamox population. We are going to be focusing for this presentation on NXRA, which is again representing some of those nitrifying populations from that second half of the nitrification process, as well as NERK and NERS, both of which are functional genes that are coding for some of the denitrification population. NERK is present in some nitrifiers, but generally we use it as a metric of the denitrifiers since it is in lower abundance due to nitrification. So again, looking at the figure here to kind of just brush up on where those different genes come into play and how they could potentially impact those nitrous oxide emissions. So some results. So the first one we're looking at is NXRA, which again, all of these graphs will have the average gene copy numbers on the side per gram of wet soil. Down on the bottom, you see the different sample locations, as well as broken up between either like the sample year or the treatments in question here. And up in the corner, there is a small reproduction of that graphic to help to keep in mind where this gene comes into play in the overall end cycling scheme of these soils. Looking at NXRA, we see some significant differences between the no-till and the conventionally tilled system here at Rosemount. We see much higher in no-till than conventionally tilled. This no fertilized treatment from 2018 at Becker is much, much lower than either of the other no fertilized sites. And if we look at Beltsville, it is hard to draw a conclusion other that the organic samples, for the most part, were lower than the conventionally tilled samples. So looking at this, we're looking at the overall gene copy numbers. We get a range of about 1 e to the 11th to slightly less than 1 e to the 12th. But that said, it is hard to compare here. There are sometimes different microbial populations between the different sites, and we wanted to try to understand what was going on with this Becker site down here. So we also looked for 16S, which is a gene that we can use to estimate the overall population, as most all microbes, specifically bacteria, should have that 16S genes. So if we look at the ratio, all of a sudden things change a little bit. Some of those significant differences we see disappear, specifically in that Rosemount site. And comparing the two, you can see that the Minnesota samples potentially had higher populations overall when compared to the Beltsville, as that ratio is now increased. The Becker site now looks a lot more comparable to the others. Something that could potentially explain this difference between looking at the ratio versus looking at the raw data here is the fact that the Becker sites, again, were archival. So part of the population could probably die off in the process of being air dried and sieved. So it is going to be a little bit more representative here, comparing it to the 16S ratio across the board. The only comparison that stayed true between the raw NXRA and the NXRA with the 16S was this conventional tilled and organic comparison at the Beltsville site. If we look at the denitrifying community and starting to look at NERK, we see some similar kind of differences between what we would see with NERK by itself and that 16S comparison. Looking at NERK, we see that the no-till was higher than the conventionally tilled again in Rosemount. We see that that Becker site from 2018 is low. We see that the Beltsville sites themselves is very little difference between the treatments there. And when we account for the 16S and the overall microbial population, we see very little differences between many of the sites, but the no-till is once again higher than the conventionally tilled at this Rosemount site. And then finally, we see NER-S. So NER-S versus, again, the NER-S ratio, slightly different things going on here. With NER-S, we see that the Rosemount site, again, is higher at the no-till than at the conventionally tilled. We have our thing with Becker from 2018 being slightly different. We also see more variation between just the Becker site from 2019 as well as the St. Paul site for 2019. And we do see some differences at Beltsville with no-till versus conventionally tilled. If we look at the ratio, though, some of these stay the same and some of them don't. So again, we lose a lot of the variability between sites. It's accounted for by just the differences in microbial population overall. That said, at Beltsville, we still see a higher ratio for conventionally tilled systems than we do for no-tilled systems. So looking at NER-S and looking at NER-K, the first thing that we see here also is the sheer difference between Minnesota and Beltsville. which prompted me to do a little bit of digging and to be able to compare potentially the niche differentiation. So early on, I mentioned that NIR-S and NIR-K have similar functions, but they do live in different kinds of environments. And if we compare, instead of the NIR-S to 16S and the NIR-K to 16S, and compare the two together, we do see that primarily looking at the actual values, NIR-S has things to the 9 to 10. So we have a range of like 2 to the 2e to the 9 versus 1.2-ish to the 10. Looking at NIR-K, it's more 2 to the 7th versus 1.2-ish to the 8th. And looking at the range from that, NIR-S is more abundant across the board. That said, comparing the ratio of the two, we do see more relative NIR-K in the Minnesota samples than we do in the Beltsville. And this could be backed up from the literature. This ratio is negatively correlated with pH. So it could be that the Minnesota samples have a slightly lower pH there. Past research has shown that NIR-K is present in both dry and wetland environments versus NIR-S is potentially only more present in wetter environments. So it could be that Beltsville is a slightly wetter site than the Minnesota sites. And this is something we hope to look at moving forward. So drawing some conclusions from all of this, we see mixed results. But we do see that these functional genes vary by management and by sample site. So we saw a relative abundance of the nitrobacter, so again, that was the NXRA gene, to be less in the organic systems than in the conventionally tilled systems, which makes a certain level of sense. So these organic systems received manure, which means that they had a slower mineralization rate potentially, which could decrease that AOB potential again, and subsequently decrease the N2O emissions potentially. Also, something to keep in mind here is that NXRA, if in these sites there was a buildup of ammonia, we could see ammonia toxicity in some of the conventionally tilled sites, which would account for the reason why, ammonia toxicity in the organic sites. If we see a buildup, we would see a decrease in the NXRA there. So this is something we want to look at, and we will hopefully have more information when we also include some other functional genes. We hope to, moving forward, also look at that AMOA gene, so looking at the ammonia oxidizing archaea and bacteria, which will help to piece apart exactly why we are seeing this sort of thing happen. We also see differences in the relative abundance of the denitrifiers. So looking at that NIR-S community again, or the ratio with 16S, we saw less in no-till than we did in conventionally tilled systems. And this was actually what we hypothesized, with the idea of we might see a greater percentage of denitrifiers. Sorry, this is opposite of what we hypothesized. We might see a greater percentage of denitrifiers in areas where we would see no-till. This is something we've seen in the past as well, looking at the differences. Finally, wrapping up, we did see that niche differentiation in the denitrification community, which again could be due to the different environmental factors that I mentioned. We saw that in no-till, there was a higher percentage of the NIR-K proportional to the NIR-S again than in the conventionally tilled. And we also again saw that the Minnesota sites had a greater ratio than the Beltsville sites. So as I mentioned, further work, we want to expand these genes of interest. We are seeing differences, and we are seeing differences by management, but trying to understand
We've lost you.
Hello?
Okay, now we can hear you again. I think we lost your audio for just a second there, Alyssa. Try it again.
Okay.
Is anybody else
Expect.
having?
Yeah, I'm also, I can't hear her. I heard her for
Okay.
just a second.
Okay, try to, you're sort of coming in and out, Alyssa, just here. We're coming down the home stretch, so.
Maybe if you stay closer, keep your body close to the microphone, maybe it's just not picking you up.
Okay. Hello. Can you all hear me?
Yes, we're good
Yes,
now.
at the moment, we're good.
Okay. Just to wrap it up, what I was trying to say is that we saw mixed results. We saw some things that we might expect. We saw some things that we might not necessarily expect. So something that might help to piece this apart some is by looking at increasing the genes of interest that we're looking at. So going back and looking at those ammonia oxidizing archaea and bacteria, we are currently in the process of trying to clone some of these standards. So that way we can get a better idea of what's going on in that part of the nitrification community, and also looking at nitrospira. So we attempted early on to isolate the Comomox nitrospira without much success. We are also having difficulty isolating just general nitrospira, so that NXRB gene. So understanding what might be going on in these systems that is preventing us from getting good samples of those populations. Moving forward, we also want to incorporate relevant nitrous oxide data collected from nearby plots at these different places. Nitrous oxide data from before was stuff that was taken historically from either these plots or things that were near to these plots, but not from these plots itself or from this sample year. So getting more recent nitrous oxide data would help to understand not just the communities that are going on here, but whether or not they are actually corresponding to increases or decreases in nitrous oxide emissions as would be predicted, and sequencing a subset of these samples. So we understand not just the functional gene abundance, but what species or genera are actually contributing to the communities that we are seeing there. And I wanted to thank everybody who has helped me along the way so far. So a big thank you to my committee, Dr. Yarwood, Dr. Cavagelli, Dr. Fiorella, Dr. DeFali. Also a big thank you to all of our collaborators in Minnesota and Dr. Rod Venteria, who has been so helpful in giving all of these samples from Minnesota and collecting that. A big thank you to the Yarwood and Baldwin lab groups, the USDA, and the Maryland Agricultural Experimental Station that has helped fund this research.
All right, we're losing you again here at the last minute, but we got pretty much all your conclusions, so that's good. Let's see if can we hear you now, Alyssa?
Yes.
There we go. We got you back. Okay, we just faded out then. We got pretty much all your acknowledgments too. So I think at this point we're going to say. No, I zoomed out. So now we're going to say, good presentation. Thank you. We're going to take some questions. I'm pointing out to folks, I just put again in the chat a link to an evaluation form for Alyssa. And so we're getting a few questions in on chat. So I'll pose those and we'll take it that way. So back on one of your graphs, Dr. Awal was asking, said there was no error bar for the Becker NF 2018 data, and he was wondering, is that because there was just a single sample?
Yes. So the Becker 2018 data, all of these samples, I'll go back to an example of one of the graphs. So all of these samples were taken from a couple different plots at each of these sites. So the error bars are showing the standard error between the different values at each of these plots at each of these sample dates. The Becker was only one archival soil sample. And while we took replicates of that soil sample, we are showing the averages between the different plots here. So there were no error bars.
Okay. Dr. Wilmot has a sort of a case, so it's a great presentation. And he has a suggestion, I guess a question he's posing, if there's enough of the 16S rDNA remaining from the samples, it might be interesting, he says, to consider characterizing it based on the overall diversity to understand the potential iron-reducing populations of bacteria. So because iron 2 that's generated during iron 3 reduction can abiotically reduce NOx species all the way down to N2O. And it would be interesting to consider if or how the iron cycle might be influencing the system.
Yeah, no, that's an interesting point to bring up. We do intend to do, again, the sequencing moving forward. And we were planning on sequencing basing on functional genes, but it might be worthwhile to also look, general 16S sequencing to see what's going on in these populations.
So as a non-microbial person, it was a lot for me to get my head around, Alyssa. But just so that I make sure that I understood, so that when you're putting these ratios with the 16S gene, that's kind of like a normalization of some sort, because just the raw numbers of replicates or whatever you generate, there's other sort of environmental factors that may drive that, even things like sample storage. Is that what you're suggesting? So it's, so would you say that the ratios are a more meaningful and more useful comparison than just the raw numbers? Is that sort of your takeaway here?
So the raw numbers are useful in that we can see the general population size between the sites. For instance, like the one that I've pulled up right here, Rosemont having the highest at its no-till site, and seeing that actual value can be good information to have to just understand the sheer size of the denitrifying population. But that said, it is very hard to compare it, especially to something halfway across the country in Beltsville, unless you were to normalize for something. So it's good information to have, but especially when you were trying to compare it, using the 16S as a ratio helps to normalize.
Okay, thank you.
Mm-hmm.
So for Dr. Wiles asking, says for conclusions that showed genes or ratios differing between no-till and conventional till, was this significant at both sites? He says, I couldn't tell from the quick view of the bar graphs whether or not that conclusion was the same for both sites.
So it was, going backwards a little bit, so it was not for both sites necessarily. So the two were NXRA here, where it was no-till versus conventionally tilled, was barely significant between the Rosemount site, but we were looking conventionally tilled versus organic was the one here, and it was only at Beltsville. The other one was also site specific, and it was only at Rosemont. So again, that's another thing we saw, was that it was variable based on management strategies, but also again that the actual sample site and the other environmental conditions that might be occurring at these very different sites could be having a strong impact on what we are seeing here.
Okay, thank you. Dr. Lansing has a question. It's more sort of related to kind of experimental design and how that was set up. But it says, and maybe you mentioned this, but she was wondering how much replication was actually being done between sites. Are these analytical reps, that is, simply splitting samples for analysis, or actually sort of replicate fields that were involved in the way the experiment was laid out? And she was also asking, are your error bars standard errors or standard deviations?
So the error bars are standard error. And as far as replication goes, it was separate plots. So we did do some replication of the actual qPCR data because there is a fair amount of variability in qPCR, in that a small air bubble can cause a very different result in CT value just based off of something like that. So we do reps of 3 on the actual qPCR plate to make sure we're getting a good qPCR read. That said, each of these samples were from different plots. So for instance, these Rosemount, they had slightly different reps, but the Rosemount had an N of 4, and those were composite soil samples, each one taken from a completely different plot. It was under the same treatment at Rosemount, Minnesota. Beltsville had an N of 3. The only one that didn't have any sort of replication would be that Becker site. That was that archival soil sample.
Okay. Nathan Sedgwick is asking about whether or not you have there were any moisture data that were collected at or around the time that the N2O sampling was done. We know that rainfall events or the amount of N2O emissions can be really strongly related to sort of recent precipitation kinds of events. And I'm just wondering, whether any of those kinds of data were collected on the samples?
Yeah, that's a great question. So moving forward, partially what we want to do is, now that we're getting an idea of some of the different functional genes that are happening here, is pairing it with some of that environmental data. So pairing it with the N2O emissions and seeing if there is soil moisture data. I personally do not have that data, but we will be continuing to reach out to our collaborators at Beltsville, our collaborators at Minnesota, to try and get some of that external environmental factor data that will probably help to make sense of some of these things that we're seeing that don't quite make sense entirely by themselves.
And I had sort of a similar comment, because you think that some of these populations, or the propensity to for the N2O to come off during nitrification, denitrification, is going to be related to sort of the overall wetness of the soil. And we know these are all cultivated, so none of them are really, really wet soils, but some may have seasonally high water tables, wetter than others, and you think that that could potentially drive that. Of course, rainfall events, those are really short term, but I would assume the populations, the things that you're measuring with your PCR, are sort of reflecting sort of intermediate length time periods in terms of the populations. The populations aren't going to change nearly as quickly as like soil moisture is going to change, right? So,
yeah.
Okay. Well, great. Well, thanks so much, Alyssa. Great job. And enjoyed it a lot. Did stretch my brain, I'll tell you, a little bit, trying to run with you on all the, I used to say that, I used to think that all this PCR and gene stuff was just kind of magic and sort of mystical. But, as I've listened to more of these, I'm getting more convinced that there's some reality to this. So thanks a lot for a great presentation.
It is still magical, Marty.
It is magical, yeah. Mystical too, maybe.
Yeah.
Is that a compliment, Marty, or not? I couldn't tell.
I'm talking about my own limitations here, Bob. So anyway, don't forget to fill out an evaluation. That link is in the chat box. And so one last question here from Ray. How dynamic are these gene numbers and ratios? If the soil gets wet and warm, will they change dramatically? Or if fertilizer is added, or are these, or are these sort of more stable? I guess the big question is, how quickly or dramatically could these change from environmental influences?
I mean, we've seen that environmental factors have an impact on microbial communities. So we would see potential changes in this. Again, you saw, as I mentioned somewhat in the introduction, increased fertilization has shown increases in some of these different microbial populations. So while the lag time might be different from site to site and might vary on environmental factors, we do see changes on these populations based on fertilization or different management strategies.
Alyssa, what are your thoughts on whether what you're measuring is a characteristic of something that reflects many years of management in this soil conditions and is sort of characteristic of that site and management system, or whether it's very temporary? For instance, if there was a flush of nitrate, wouldn't within hours or certainly a day, the populations of certain bacterial communities go much higher? And if you sampled at that time, you'd get a very different answer than if you had sampled, say, before that nitrate episode or before a rainfall or that kind of thing?
I mean, again, I do think it has an impact. We tried when we were sampling here to sample at a very similar time and during similar seasons.
I understand there's seasonality, but
yeah.
So the
actual
denitrification certainly, dramatically changes is episodic, right? So if you had a warm, wet period, the products, the activity goes up dramatically. Does that also mean that your gene numbers would go up dramatically?
No, not really. So not with the DNA.
What you're saying is the organisms are there.
Yeah.
It's not that So the response, let's say the response to rainfall and warm conditions and nitrate, and you get this big peak of N2O,
Yeah.
that is not a organism multiplication. They're not growing, but they're just activating.
They're
those
activating.
there. Okay. So
Yeah.
over what period of time would they be growing?
I mean, they
And
can
if you
grow.
give more substrate, certainly the population is going to increase, right?
Right, but we know that growth rate of the microbes out in the soils are not really very reflective of like lab-based studies, because we've grown really, frankly, a lot of weeds in the lab. So where we see a lot of, increases in populations within the lab, we tend to basically see the numbers, population shifts to be relatively small comparatively, like in the soil system. And part of that is, I think, spatial isolation. That leads to that. And so what you will see a big shift in is RNA. And so we did some work early on with Holly Bowen when she was here, inducing denitrification, and the RNA shifts. Yeah, and Jude was going to comment because he's also done some of that work.
Yeah, I was just going to say, when we've done some similar work in FSP over the seasons and in short time courses, and you see an order of magnitude change, but it's more sort of like a seasonal response. And the doubling time of a population of bacteria in the soil, depending on the conditions, might be 7 to 10 days in terms of when they've used C14 to do feeding studies and pulse chase experiments. Sometimes it's much, much slower. You get 2-week doubling times for microbial population. So it's really kind of like a monthly type of response in terms of microbial biomass.
So that sort of conflicts with some microbial biomass studies. And if you start looking at, say, bacterial feeding protozoa or something like that, they tend to be much more responsive to change of conditions. So how much do we really know about how fast microbes, or particular microbes, how does the 16S change, for instance, as the soil warms up in spring? Does it change?
Yeah, that's a really good question, because copy number can change, and we're only now getting a handle on that, but but linkages between total microbial biomass and reproduction rate have been done with feeding studies. But you're right that you could have massive changes, and we don't know also because you have archival both DNA and DOC that get caught up in those types of studies. So I think you're right. There's lots of loosey-goosey stuff in there, but as far as we can tell, in the soil, the conditions are really not superb for microbial.
Right.
This big these big growth rates. Yeah.
Right. This is a great discussion, but
Yeah.
we're going to pull this to a close. Thank you again,
Okay.
Alyssa.
Yeah, sorry to keep it
Great
open,
job.
but we don't get to kind of sit in the hallway and.
I know.
Yeah,
Well,
Thanks, Alyssa.
But we'll consider this the hallway. You can stop the recording. Yeah, it was
Thanks.
nice
Thanks
job.
very
I'm
much,
sure you
everyone.
learned a lot in this lesson.
Good job, Alyssa.
Thank you.
Great.
Don't forget to fill out the evaluation. Great.
Yeah.